Welcome to the Lymphoma Lab at Aiims New Delhi. It was developed by Dr. Soumya Ranjan Malik, Professor, Department of Pathology, and currently has numerous members, including PhD students, scientists, research scholars, and technicians. Today, we will demonstrate one of our routinely done techniques of T cell receptor clonality testing for detection of neoplastic T cell population. For today, we are using formalin-fixed paraffin-embedded tissue specimens. First step of our procedure is DNA extraction, for which we will prepare 5 slide sections of 10 micron each by microtomy. After trimming, the ribbons are cut and then floated on a warm water bath, after which they are picked up on a slide, followed by complete air drying. We use slight scraping of the tissue only to avoid excessive wax contamination. The tissue which has been scraped is then put in an Eppendorf tube of 1.5 ml. We are using Promega Relia Prep system and DNA will be extracted using the manufacturer's protocol. For deparaffinization, we will add 300 microliter of mineral oil into the Eppendorf tube and incubate at 80 degrees centigrade in the dry water bath for 1 minute. For proper mixing, we will vortex it. In order to lyse our tissue sample, we will add 200 microliter of lysis buffer, followed by centrifugation at 10,000 G for 15 seconds. Here, we will see two phases, the upper oil phase and the lower aqueous phase. The aqueous phase is which we will proceed for the further procedure. In order to degrade the protein, we will add 20 microliter of proteinase K and mix by pipetting. After which we will incubate at 56 degrees centigrade for 1 hour, followed by 4-hour incubation at 80 degrees centigrade. This step is crucial to reverse the protein DNA and DNA-DNA crosslinks which are formed during the fixation of tissues. After the incubation, allow the sample to cool at room temperature and centrifuge briefly to collect any condensation. To degrade the RNA, add 10 microliter of RNase A to the lower phase and mix properly by pipetting. Incubate this mixture at room temperature for 5 minutes. After that, 220 microliters of BL buffer or binding buffer is added to the lyse sample. Followed by 240 microliter of 95 to 100% ethanol and thoroughly mixed by vortexing properly. Centrifuge this tube at 10,000 G for 15 seconds. Here again you can see two distinct phases. One, the upper oil phase which has all the wax, and the lower blue aqueous phase. Now transfer the entire lower blue aqueous phase to the binding column or the collection tube assembly and cap the column. Centrifuge at 10,000 G for 30 seconds. The flow through can be discarded at this procedure and reinsert the binding column into the collection tube. For column washing and elution, add 500 microliter of 1x wash solution with ethanol added to the binding column. Cap the column, followed by centrifugation at 10,000 G for 30 seconds. This step is repeated 2 times. After this, open the cap of the binding column and centrifuge the binding column collection tube assembly at 16,000 G for 3 minutes to dry the column completely. Transfer this binding column to a clean 1.5 ml microcentrifuge tube with proper labeling and discard the collection tube. Add 30 to 50 microliter of elution buffer or nucleus-free water to the column and cap the column. Centrifuge this at 16,000 G for 1 minute. Remove and discard the binding column. Cap the microcentrifuge tube and store the eluted DNA at -30 to -10 degrees centigrade. We are using nanodrop spectrophotometer for DNA quantification. In this, we will add 1 microliter of the eluent or NFW to be taken as the blank, followed by 1 microliter of the eluted DNA. We will be seeing a good graph of the extracted DNA with a good 260-230 and 260-80 ratio. For step 2, that is clonality, we are using T-cell receptor gamma gene rearrangement assay 2.0 by in vivo scribe. This is stored at -80 degrees centigrade. The kit includes ladder, positive and negative controls and master mix, which we have already aliquoted. The PCR reaction should be prepared in the hood, and for every reaction we should have tubes for a case, the positive control, the negative control, the NTC and the ladder. This is the TCR gamma clonality master mix. This is the ladder, which will give us an approximate size of the DNA fragment size, followed by positive control, which will give oligoclonals or clonal peaks. This is the negative control, which will give a polyclonal peak, and this is the Amplitech Gold polymerase. For a 50 microliter reaction, we will add cocktails of master mix, DNA tag polymerase, and the template DNA, that is the case DNA, positive or negative control DNA, and no template controls. Vortex and briefly centrifuge the tubes. For the PCR reaction, we are using Applied Biosystems Ferrity Thermal Cycler, and we will set up the PCR program including steps of denaturation, annealing and extension for a total of 35 cycles, followed by final extension at 72 degrees for 10 minutes. To confirm the success of our PCR reaction, we will put up an agarose gel electrophoresis. We will prepare a gel of 2% agarose followed by casting, followed by electrophoresis run. The gel is then captured and here we can see good products. Now we will proceed to the final step of fragment analysis. To each reaction mixture, we will add a combination of Liz and high dye. And we will prepare our plate accordingly. Heat denature the samples at 95 degrees for 5 minutes. Followed by snap chill on ice for 5 minutes. We will be performing the fragment analysis on ABI 3500. This is the capillary. After switching on the system, we will click on the 3500 application and set up our reaction plate as per instructions. For our experiment today, we will set up a fragment analysis and after appropriate checks of the system and the materials, we will then load the plate into the instrument, and after checking the pop, the anode and the cathode buffers, we will start the run. After the run is complete, we will get results in this format. For every sample, we will have a ladder, which we can see in the Liz peaks in the orange bars. Then we'll have the positive and the negative control, and the sample itself. Thank you for listening.